this question would be good for you to answer with your background. Ethan asks, how can we move away from the process is the product to the product is the product, and thereby allow for iterative innovation on products that work but need to be made more efficiently?
The product is the product. You mean changes to the product? Yeah, I mean, from a regulatory perspective, right? If you're in an IND stage and you're making manufacturing changes, you can continue to, if you provide supporting data, you can continue with the same IND and move to a new version. But if you're changing the product from a regulatory perspective, you would have to essentially start a new clinical investigation under a new IND. I think the process is the product is the more traditional way to think about it, but I think we moved away from it for quite some time now, because that paradigm is basically, if you can show me that the process is consistent, then I will know that the product is. But really, we're asking to show product quality data to support changes these days, and just showing that the process performance is equivalent is not going to be enough. So I think we kind of moved away from that. We just like to say it every now and then, but from an FDA perspective, we moved away.
I think that's critical. I'm glad Ethan brought it up, and Tal can speak to it with such authority. Maturing cell therapy to a point where we have enough process and product understanding to rigorously define critical quality attributes is going to be an absolutely essential part of the field maturing. That's where we are with antibodies, where, okay, we can make process changes and we understand whether sometimes we make process changes with antibodies and the product, there are some changes, but we understand the clinical significance of those changes and say they don't affect clinical performance. Or we understand that, hey, we did change this one thing and it could change the PK or the half-life or the immunogenicity, and then we stay away from those. And that's where the cell therapy field, an important part of this like accelerating innovation, is the ability to define our product, what are the critical quality attributes, so that we're not, I think the other thing that's handicapping the field today is that we don't have this understanding. And so then we don't want to make process changes because what if it does change the product? It could even be, and I think this is almost the more important point, it could even be that we could, I don't think the bar is, can we push it through FDA? I'd be interested in what Tal would say about this. Let me poke you a little bit. I don't think the bar should be just, hey, can we get comparability so we can push it through FDA? It can even be like, hey, we're concerned that if we do this, we might demonstrate comparability, but we don't have enough analytical understanding of the product, and it may actually change it. And do we want to take that risk in product development or not?
Yeah, and I think there is a difference if you're making manufacturing risky manufacturing changes like that at early stage versus later stage. At later stage, you are expecting to have some understanding of how these changes would impact safety and efficacy. At an early stage, you just don't necessarily have that knowledge. And when I say manufacturing changes, I mean, there are manufacturing changes outlined in some of the recent guidances that are not considered manufacturing changes, but they would be considered a change to the product, like changes to sequences of vectors and things like that, where FDA just doesn't like to see that, again, because the uncertainties around it. But I think, again, you would get much more flexibility of process changes at early stage, much less scrutiny unless they have a potential to impact safety.